All right, good morning, afternoon, or evening to all of you, wherever you are in the world. My name is Eugene Yaboa. I am a senior specialist in next generation sequencing and also a specialist in bioinformatics. This is long due, but here we are at it again. The goal for this training is to develop and help students acquire the knowledge needed to analyze any sequences, regardless of the instrumentation. So this will be either Illumina, Oxford Nanopore, the ion torrent instrumentations, any of them. And the goal is to be able to take a FASTQ file from any sequencer. And this could be either SARS-CoV-2, if you have to assign lineages, flu, influenza, if you have to subtype, metagenomics, differences between the assembly types, de novo assembly, or reference-based assembly. We are going to go through all of that. I recently had a training where about 30 participants, I asked the participants if they had previously taken any bioinformatics courses. And 28 of them said yes. Some of them had even taken numerous bioinformatics trainings. So before the training, I provided them with 2 files for SARS-CoV-2 on the Illumina instrument. And I asked them to tell me what the lineage or clades were. And none of them could accomplish this task in 10 minutes. Some didn't even know where to start. Actually, the majority didn't know where to start. So this called for the need to not only teach people bioinformatics, but actually train them in the concepts so they can be independent. So this is my goal for this training. It's not to show you how things are done, but to actually teach you how to do these things and understand the fundamentals, the concepts, and everything attributed to the workflows that we are going to be using. After this training, and there are going to be a series of them, the hope is that you will independently be able to take any FASTQ files without needing to email me for assistance. You actually gain the skills to troubleshoot any errors by yourself. That is the hope. And I think we'll be able to achieve it with the new method of teaching that I have developed. In this training, we are going to utilize the software called Genius. So I am going to walk you through how to get the free version of Genius Prime. And Genius Prime is what we are going to use initially for at least the first 2 sessions to get you up to speed with actually what bioinformatics tools are and how those tools are utilized. That should give you some basic foundation. So when we build up on that, you have a solid idea about what tools and bioinformatics applications there are available. So to go straight into it, we are going to go to Google first. So any web browser will take you there. Once you are on Google, we are just going to type in Genius, Genious, Prime free trial. And usually it's the first one that comes up. On the free trial link, I just click on it, and it brings me to the Genius free trial website web page. You can see in the middle of the screen, it says start your 30-day free trial of Genius Prime. You put in your email and you click get started. So complete your profile. To complete your profile, it asks you if this is your first time using Genius Prime. If it's a new email, you choose yes. Which of the following best describes your current role? Academic, government, student, the country you are in, United States of America. It could be any country you are in, and you click get started. You get to this page where it asks you to download and activate your Genius Prime. So the first thing you do here is that you download the Genius Prime software. You install after you've downloaded the Genius Prime software, and then you sign in. So once you sign in, it will give you the option to put in your activation key. So once you have downloaded Genius Prime, the activation code will be sent to your email, the email that you registered with. Mine hasn't come in yet. It usually takes a few minutes to come in. But once you've downloaded Genius Prime, you are still able to use Genius Prime, but some features are restricted. So you can open up Genius Prime after you have downloaded the software. And I'm opening mine. It looks like this. And it's usually very fast. So once you download it, you will get a screen similar to this. I have a lot of work already populated in my left pane, but you probably have nothing in your screen, which is fine. So on this section, I use the marker. So this side, I have a lot of applications that I've already ran, and you may have nothing on yours. If you start using Genius Prime right away, you are going to face some restrictions. So the best thing to do once you open the software or the first time you open Genius Prime is to go to Help. Under Help, you will find Activate License. Remember, you have the license key for your free trial emailed to the email you registered with. So once you receive that email, go into the Activate License from the Help section, and then you will have three options: license key, license server, or email sign-in. You can choose the license key because that is what is going to be given to you once you sign up. And it will make sure you choose the right email. And then your license key, you just copy it from the email they sent you and then paste it in here and then click activate. This will activate your session so that now you can use Genius Prime fully for the next 30 days. All right, so we are going to stop here with Genius Prime now that it's installed and ready to be used, and we'll get back to the applications on how to navigate Genius Prime and use Genius Prime. So we just learned how to and walked through how to install Genius Prime for some of the bioinformatics applications and work that we are going to be doing during this training. The next tool that you should definitely have and definitely get is called Terra.bio. So we are going to close this session for now, and then I am going to walk you through on how to get the Terra.bio account set up and how to navigate the Terra.bio platform. So what is Terra.bio? It is a graphic user interface for that hosts applications, pipelines for bioinformatics. It's also a collaborative space. And one of these sessions, I am going to invite the team that built or host the Terra.bio platform to tell you more about the platform, its capabilities, and the things that are in development to support bioinformatics, not only for public health, but for like other very numerous applications. If you have a Terra.bio account already, you do not have to go through the process of setting a Terra.bio account up, which is quite tedious because you need to create a billing that is linked to your project. There is a promotion sometimes where you can qualify for free $300 credit to apply to your account. We are going to go much deeper into how to set up a new Terra account for new users and how to activate this service. Those of you who already have a Terra.bio account, I am going to show you how to utilize the Pathogen bioinformatic resource page. Once you understand how that page works and how that page functions and what is actually in those pages as resources to analyze your genomic data, you can do a lot with that platform. Whereas if they actually understood where the inputs are coming from, how the resource page can actually help them, they'll be able to troubleshoot these issues and these errors by themselves. They will know where to get the data and the information that they need to run any workflow by Pathogen. And that is the goal. The goal is to get every trainee to be independent and actually understand why they are doing what they are doing, rather than them just following along and following a protocol, but without understanding the fundamentals of why they are putting in the numbers they are putting in or clicking the buttons they are clicking and what effect those do if they're not done correctly. So that is the primary goal to walk you through the Pathogen resource page if you already have Terra.bio account. A lot of institutions now are using Terra.bio, so most people already have it. If you don't have Terra.bio, don't panic. Don't let your heart be troubled. I will walk you through how to set up a Terra.bio account in the next video. So for now, let's go on navigating the Terra.bio account, but specifically going through the Pathogen resource page, where we can find all the necessary information we need to run any pipeline or workflow designed by Pathogen for public health bioinformatics. So we go back to our friend Google and we type in Pathogen, Theaiagen, genomics. Hit enter. And we look for Pathogen Genomics. And there it is. Pathogen Genomics. So we go on the web page. And on the web page for Pathogen Genomics, we are looking for resources. That's because we want to learn about the resources that they provide. So we go to resources. They have what we do, industries, about, contact us. We want resources. So on the resources, there are three options. There's the news, public health resources, and newsletter. We go to public health resources. The public health resource page has a lot of information. So follow me closely. I'm going to scroll to about the middle page. So our goal here is to do bioinformatics. So we find public health bioinformatics. And they have different versions from version 0.1 all the way to version 2.1.0. We always want to go for the latest version. If I move this here, we are always going for the latest version. And in this case, it is version 2.1.0. So we click on version 2.1.0. And that brings us to the public health bioinformatics section, version 2.1.0. I'm going to scroll all the way to about the middle piece part, and I have a couple options. On the far left, I have all workflows, all SOPs, guide to phylogenetics. There's just like a lot of resources. I'm going to go to all workflows. So under all workflows, I am actually able to see every single workflow that has been designed by Pathogen and is available on Terra.bio to analyze genomic data. Like I said before, usually when people are going through Terra.bio training, it is so pathogen specific that there is actually not enough time to walk people through the resource page. So we train people on how to analyze HIV using a workflow for HIV. And then once we leave, they can't do anything else except HIV. And when they are faced with a public health threat or problem, or the ministry asks them to analyze a different pathogen, they are stuck and feel like they can't use Terra.bio. But that is being changed. So my goal is to change that, and instead of teaching an and training on specific workflows, I'm actually going to train on the background and the resources that Pathogen offers so that participants can actually run any workflow they want. So it's more like I am giving you the ingredients so you can make your own food rather than giving you the food. That way you can also be creative. Because once you understand how these workflows work and what is needed to run them, if you encounter any issues or something breaks, you'll be able to fix it, or you have an idea on what is going on and how to fix it, rather than just running workflows blindly and then having to resort to emailing the trainer when you encounter issues. So let's go through some of the workflows that they have and how to actually use it. So the first one is SRA fetch, assembly fetch, and it has the name, the description, and what taxa it can identify. And this is grouped by kingdom. If you're looking to do just a really general genomic assembly, QC and characterization, there are 6 of them. There is Theakov workflow. There is, and that is for viral genome assembly, quality control, and characterization from amplicon sequencing. And the pathogens that you can use this workflow for SARS-CoV-2, for monkeypox, for HIV, for West Nile virus, for influenza, for RSV-A and RSV-B, and all viral. So if you have any of these pathogens, or you're looking to categorize, characterize, and identify the species, you have the Theakov workflow for all viral sequences. Theaproc workflow series is also used for bacterial genome assembly, quality control, and characterization from whole genome sequencing data. And this is for bacteria. So if you want to analyze bacteria data, you'll be using this workflow. Go down the list. Thea-Uc is for analyzing mycotic genome assembly, quality control, and characterization from whole genome sequencing. And the pathogen identified to identify mycotics. Thea-Meta is genome assembly and quality control from metagenomic sequencing. So all of you doing metagenomics, if you're looking for a way to analyze your samples, Thea-Meta workflow is your best friend. And then we have Pangolin update, this Vader update. And then this is for SARS-CoV-2 metagenomics in wastewater. You have the Freya wastewater analysis series, and that recovers relative lineages, abundances from SARS-CoV-2 wastewater sequencing data, and it will also generate visualizations. So that is for SARS-CoV-2 and can also be used for some viruses. For those of you interested in genomic epidemiology or how to build trees and interpret and construct trees, you have workflows built in for you as well. So there is the Augur workflow, and that is for phylogenetic analysis for viral pathogens, so all viruses. The core gene SNP, CZ-Gen Epi, this, the PrEP, I am not sure if this is still running, but I know the CZ-Gen Epi platform is now being it's being navigated and taken over by Pathogen Genomics. And I think in November there's going to be some changes where they'll actually take over and migrate the platform from CZGen Epi, the original platform, to a new Pathogen platform. But more on that to be discussed. If you have an account with CZGen Epi, you should have gotten some communication or some email about migrating your data. KSNP 3 is for SNP-based phylogenetic analysis from assemblies. So there's a lot of things to build trees. Public sharing data. There's just a lot of information. So it's most people using Terra actually aren't aware of these great resources that the platform that Pathogen has to analyze data. There is just a lot of information. So now, how do you use it? The way that you navigate. So let's say, example, I have a SARS-CoV-2 sample that I just sequenced, and I want to analyze the data. How do I start? So first, I come to the Pathogen resources page, and I'm going to go back, and we are going to redo it. So you're going back to Google. We go to Pathogen Genomics. We look for Pathogen Genomics. Under Pathogen Genomics, I'm just going to accept the cookies. I'm looking for the resources. So I look for resources. And then under resources, because I want to do public health resources, I don't want news or newsletter. So I go to the public health resources. Under the public health resources page, I'm going to scroll maybe halfway. And I want to do bioinformatics. So I'm looking for public health bioinformatics. Something to note is that there are a lot of different versions that has been created and developed over time. You always want the latest version. In this case, it's version 2.1.0, the top one. So I click on version 2.1.0 for the public health bioinformatics. Now this brings me to a page where it shows all the workflow documentation. For now, we are not going to worry too much about that. We are going to go straight all the way to about halfway. We have options for, on the far left, all workflows, all SOPs, guide to phylogenetics. We want the workflows because we want to figure out how we are going to analyze our SARS-CoV-2 samples that we just sequenced. So on this page, once again, we are looking for how to analyze SARS-CoV-2 samples. We just go through the workflows and we look for the one that fits what we want to do. So here we can see that the genomic assembly and characterization tools, our workflows that we have, the first one is TheaCov workflow, and it's for viral genome assembly, quality control, characterization from amplicon sequencing. And when we scroll down to on the right to where the taxa is, it tells us that this workflow can analyze SARS-CoV-2, monkeypox, HIV, West Nile virus. So since our sample that we want to analyze is SARS-CoV-2, we are going to use the Thiakov workflow. So similarly, if you had any bacteria, you are going to use the Thiaproc workflow. So that is for bacteria. And this actually breaks it down for you. So you don't even have to guess. You look at what you're trying to enter, what pathogen you are looking for, and then you pick the workflow that suits that question you are trying to answer. So to click on this Thiakov workflow, once I put the cursor, I see an open. It opened as a page, so I'm going to click on the Thiakov workflow open as a page. So now I am in the Thiakov workflow series, where it tells me that it can analyze SARS-CoV-2, monkeypox, HIV, West Nile virus, influenza, RSV-A, RSV-B, and all viruses. That is what I need for my SARS-CoV-2. It has the description. It has the code on GitHub. It has the doc store where you can actually pull it into your workspace. And command line if there are command line applications or compatibility, and it's yes. And then you have some option features. So once again, the Thiakov workflow series will be able to analyze all of these SARS-CoV-2, monkeypox, HIV, and all the other viruses. Once we scroll down, so we are looking for inputs. So for bioinformatics, you usually have specific inputs, and that helps identify your sample that you are putting in and some characteristics about that sample. And then there's also specific output that you may need from the data. So example, if I'm putting in SARS-CoV-2, at the end, I want to know the Pango lineage. I want to know the clade. I want to know if my sample is good quality. So my output, I can select next the clade, because I want to know the clade. I can select the Pango lineage. So that is usually how it works. You put in an input, and then you specify the output. Pathogen has made it very, very easy to navigate and to know what inputs you need and what outputs you need, so that you don't have to guess. So it takes the guessing out of the learning, which is very, very, very important. So once again, for my previous example, I have SARS-CoV-2 samples from the ministry, and I'm trying to identify what lineages they are. So I went to the Pathogen resource page, and I found out that when I went to the workflows, the Thiakov workflows was the one that can analyze SARS-CoV-2 samples. So I picked Thiakov, and now I am looking for the inputs and outputs. So those are the two things that you'll be looking for. This has a lot of information regarding the workflow availability, the overview, the core tasks, the input, organism-specific parameters, a lot of information. My goal now, or at least for today, is SARS-CoV-2. So what I do is I scroll all the way down to inputs. So inputs, there we go. I go to inputs, and I pick which instrument I'm using or what instrument I use to sequence my samples. So if it's Illumina paired end, these are your inputs. Illumina single end, ONT, Oxford Nanopore Technologies, the Clear Labs, it's here. They don't have it for the ion torrent. So if you have that data, there's a different way to analyze those. But the ion torrent, there's a plugin that actually does your analysis. Generate consensus will actually do your analysis and assign lineages. So that's already built in the ion torrent sequencing system, where you don't need to pull your FASTQs files and do lineage assignment outside the instrument. You could all do it all inside, and specify. So as it runs, you specify the output workflow in the ion torrent, and then it will just run the whatever that workflow is that you want. In this case, your SARS-CoV-2, and it will generate the consensus, and it will do the alignment with a reference, and it will give you QC parameters, and will assign the lineages as well. So we are using Illumina paired N. So we click on TheaCove Illumina paired N. We are still in our input. And then it gives us some more information about what we need for the input. Let's see. Because TheaCove, like we said before, has a lot of different applications for different pathogens. So the TheaCove workflow is used for SARS-CoV-2. It's used for flu A, it's used for RSV. It's used for HIV. It's used for West Nile virus. So there are different inputs for all of these organisms, so that you are putting in the right input for the right things that you are analyzing. So because of that, when we go to input, we'll just scroll down to where it says organism-specific parameters, because there are many organisms that we can analyze. And we click on the organism-specific parameters. And under the organism-specific parameters, it gives us all the parameters we need for the input. So for SARS-CoV-2, and you can see it says organism SARS-CoV-2. If you scroll down, this organism monkeypox. If you're doing monkeypox, you can load more. You can go down. There's West Nile virus. There's flu. There's RSVA. So they'll all be available. So what we are going to do is, for SARS-CoV-2, these are our inputs. And these are all specific parameters for our inputs. So on the next video, we are actually I'm going to show you where to put these inputs and how to run the analysis based on these inputs. There are also some, so click on SARS-CoV-2, let's bring this down. And I did mention outputs. For output, you can just click output here, and it will tell you TheaCove workflow will output results in a set level, which is fine. Because you, once again, they've made it so that you don't have to guess on the output. So you pick use default for all your outputs. And then once the output is executed, at the end, you just pick whichever variables you want to look at. So for now, this is how you navigate the resource page once you have a question in mind and a pathogen that you want to analyze.
Don't forget to hit the like and subscribe button below so you can get notifications when new videos are uploaded.